All right, hey, thanks for coming to today's version of Micro Seminar. It's the first one in a while, and I just want to give a plug. If anybody's out there is interested in giving a micro seminar, just please contact either me, Dave Baltris, Lizzie Wilbanks, or Cameron Thrash. We're trying to schedule things for kind of April on, and so ton of stuff open. Just please contact us. We'd love to host you and love to have you share your science with us. So it's my absolute pleasure today to actually introduce Dr. Katherine Milligan-Myhre. Myhre, is that it's Myhre? Right. All right. So first, I want to just give you a huge thank you from all of us, because you've been doing some amazing work the last couple of days, just kind of internalizing and giving out a lot of support to a lot of grad students out there. So just from me, and I think from a lot of people, just thank you. I'll link to the thread, and I'll link to the all of the advice and all of the situations that you've shown. I'll link that in the YouTube comments. And so if anybody hasn't taken a look at that, just please take a look in terms of grad school awareness and grad student awareness. It's really important to kind of support people, and Kat's done a really awesome job relaying people's stories over the last few days. All right, so now that I've said that, it is my absolute pleasure to introduce Dr. Katherine Milligan-Myhre. Kat was an undergrad and a grad student at the University of Wisconsin-Madison in Laura Knoll's lab for graduate school. And then we actually share a background. So Kat was then a postdoc in Karen Gilliman's lab. Karen Gilliman was one of my PhD advisors. And Kat was also co-advised by Bill Kresko at Oregon. So Bill was actually my first rotation in grad school was under Bill. So I was working with sticklebacks way back when. And so it's awesome to see kind of how the system has blossomed and thrived over the last few years, since 20 years or so since I was actually working in it. Today, I think Kat's going to talk about sticklebacks and microbes. I think, sorry, there's a lot going on here. I got bee guys outside and stuff. There's a lot of activity. So, but aside from that, please take it away, Kat, and we're looking forward to hear what you have to say.
Thank you, Kuyana. So, ubla ubana ataga napaktak miuruna, kititagak miuruna, nipak miuruna. Apaga James McClellan, Anaga Sally McClellan. So if you see them, tell my parents, Sally and James McClellan, you heard me on the web. They'd be super psyched. So what I said is, Ubla Lota, good morning, good day. Inipak Miaruna, I'm Inipak, Kititagak Miaruna, I'm from Kotzebue, Napaktak Miaruna. My people are from the Napaktak Miuk tribe in Northwest Alaska. And I'm currently, as Dave said, an assistant professor at the University of Alaska Anchorage, and I've been here for 3.5 years. So I'm going to do a quick share, which of course I knew how to do a minute ago. Here we go. Entire screen. You're going to see yourselves for a second. Let's see, are we sharing?
Not sharing yet.
Okay, there we go. Okay, so now you should see stuff. So what I'm going to, I want to start with saying land acknowledgement. So thank you to the Denai people whose land the University of Alaska is on and who are still here. So just want to acknowledge those people. So as Dave said, I'm going to be telling you about the work that I do adapting an evolutionary model for host microbe studies. And this is work that I got into as a postdoctoral fellow at the University of Alaska Anchorage. And I was really lucky. So when I was a grad student, I studied pathogenic microbes, and I did that as an undergrad as well. And so I had all this experience in microbes that can do harm. And that's what a lot of people were studying for a really long time. We've been studying microbiology for hundreds of years, but it's always been in terms, or for a long time, it's historically been in terms of how are microbes damaging us. And that was really important when I was an undergrad and when I was a grad student. But towards the end of my grad program, we started thinking about not just how we can conquer these microbes, how we can kill these microbes, but also how we benefit from microbes. And it turns out that these microbes are not just trying to kill us. It's not all microbes are trying to kill you. And in fact, a lot of microbes live symbiotically with us. They don't really know what we are, right? But they live with us, on us, all over our bodies. They're on our skin, they're in our eyes, they're underneath our armpits. They're part of the reason our armpits smell. You know, they're just a major part of our body. And these microbes are not actually trying to harm us. They're not trying to force our cells to break open so they can extract nutrients. They're not producing toxins to kill us. But instead, what they're doing is really great things for our body. They're stimulating our immune system. They're stimulating our growth. They're stimulating brain development. You know, these little tiny microbes that are associated with your body at some point in your lifetime stimulated a cell which sent a signal all the way up to your brain to say, hey, you need to kick in and start developing because we need this host to survive. So not saying that the microbes thought that, but the result of all these interactions help stimulate our development and our growth. And they also do things like make things that we can't make, like vitamin K. Now, in our lab, we're specifically interested in the microbes in the gut. I really like the microbes in the gut. All the things that I just told you are things that those microbes can do. And in fact, another thing they can do is they can prevent microbes that would cause harm from establishing, colonizing in our body. And so they're really important for maintaining our health and helping us develop. A lot of work has gone into, a lot of beautiful work by a lot of amazing labs has gone into really defining who is there, describing which microbes are present in healthy people and which microbes are present in ill people and which microbes are present in the very early stages of our lives and which microbes are present when we get older and how those microbes are changing over time and with their diet and et cetera. But what I'm really interested in is taking that information of who's there, but then also figuring out what happens when you disrupt those microbes? What happens if you kill some of those microbes off? What happens if you are exposed to contaminants that allows some microbes to grow and kills others? So we do this in our lab. And we're also interested in this relationship between the microbes in the gut. So here I have my beautiful gut that I drew during a seminar, much like this one, given by somebody else. And this is a pretty cool cell layer because I'm just showing it as this little homogeneous cell layer, but there's actually lots of different types of microbes there. And that's something that I studied when I was at the University of Oregon. And there's also this really nice mucus layer, and that's being studied by a lot of great labs, including my former postdoc lab, Karen Gilliman's lab. But what I'm really interested in is who's here in the gut microbiota. This is the lumen of the gut. It's basically a tube, and there's a ton of microbes there. And then how are they interacting with the cells that are in the gut and these immune cells? So this fabulous drawing is of a dendritic cell that's sending a little part of itself through the cell layers to get a taste of which microbes are present in the gut. And then they send a signal inside that says, hey, we need a lot of reinforcement here, and recruits a lot of immune cells, or says, hey, everything's good, and suppresses inflammation. That's no oversimplification, but hey, I've got 25 minutes, not a whole semester. Okay, so this relationship is complicated because there's lots of different cell types, there's lots of different microbes. And then just to complicate things a little more, there's a bunch of genes that are involved in all this too. So you don't have to memorize these genes. I don't memorize these genes. You don't have to memorize these genes. But these are all the genes that are, or some of the genes have been identified to be involved with Crohn's disease and ulcerative colitis and inflammatory bowel diseases in general. And I'm not showing this to freak you out. I'm just showing this to show you that there are a lot of genes that are involved with this complex interaction between microbes and their gut. So what's cool about these diseases, or horrible if you have them, is that they're all characterized by a change in the microbes that are present in the gut and that microbial community and the immune system in the gut. So these diseases are all associated with inflammation in the gut, so an increased presence of immune cells in the gut. And that's also also followed with or associated with painful inflammation, pain in the stomach, change in bowel capabilities, etc. So it's this complicated relationship. And the reason I like to show this image is because there are so many different genes. You know, if you were going to try to study this in a mouse and you wanted to look at the immune-associated genes, let's say associated with Crohn's disease, you'd have to be able to knock out or knock down all of these genes. So what we've done is we've taken a system three-spine stickleback that has a lot of genetic variation, and that just means that there are a lot of genes that are different from one population to another, and I'll talk about that more in a minute. But that's how we get around having to make a knockout out of every single gene in our little host organism. So our lab is really interested in host microbiota studies. We're interested in the inflammatory state and the microbial community, and we do this by making germ-free fish. Now, back in the day, people started looking at, these studies have been going on for a long time. So these are images from Life magazine from the 1950s showing how they would make germ-free animals. So here we have a pregnant rabbit, and she's giving birth in a pool of disinfectant to reduce the number of microbes. And look at this, he's not wearing gloves. This kills me every time. And then here we have another image of somebody who was completely covered head to toe in equipment to make sure that his germs don't get on these animals. And I like showing this for two reasons. One is because I don't have to wear this getup, so we've come a long way since the 1950s. But two, because I like showing this because the animals that they're actually using here are not inbred animals like we typically use now. They have genetic variation. And that's what we're really interested. Because we're interested in this really complex relationship between the inflammatory state and the microbial community and the host genetic background, we like to use animals that are not inbred. So what are we asking in our lab? In our lab, we are interested in how does the host genomic background contribute to host microbe interactions. And we're using this cool little fish here, three-spine stickleback. We also occasionally play with nine-spine stickleback. And yes, to that person in my inbox, I know I need to get you some nine-spine stickleback. But we use this animal three-spine stickleback. And one of the main reasons that we use this organism is because it has a lot of genetic variation. And in fact, the genetic variation between one population of stickleback and another population of stickleback is the same as genetic variation between one population of humans and another population of humans. So for all of you NIH program officers listening out there, it's a really great model for host microbe interactions that reflect humans, because it has the same, this organism has the same genetic variation as humans. We are located here in Anchorage, and in Anchorage we have access to anadromous fish, and you're going to hear about this a lot. So it's basically a fish that lives out in the ocean, and then it comes into freshwater to lay eggs. And then those eggs turn into juveniles, and eventually those fish go back out into the ocean, spend most of their time there, and then come back in. So anadromous just refers to living part of its life in the ocean and part of its life in freshwater. And then we also have freshwater populations. And these are populations of fish that started out as anadromous. So anadromous are the ancestors of freshwater fish, but then they got caught up in this fresh body of water somehow. Maybe a glacier receded. Maybe they were picked up in a net and dropped off in a lake. In whatever case it may be, they had to adapt to this new environment that was, they were in a high salinity, now in a low salinity. They had to adapt to new predator, new prey, new food sources, new pHs, new chemistry, water chemistry, just lots of different things they had to adapt to. And they did that all over Alaska, all over Oregon, Japan, Netherlands, just all over the Northern Hemisphere. And I'm just focusing in on Alaska because that's where we are, and those are the fish that we play with. But if you go to a stickleback conference, you get to hear about people using fish all over the Northern Hemisphere. Now here we are in Anchorage. We happen to be within driving location of a lot of lakes, and I like showing this one because this shows you all the little blue dots, and those are all lakes that we have access to, and a whole lot of those have stickleback fish in them. So what we tend to do is we drive from Anchorage to grab some anadromous fish from somewhere in here. Sometimes we get them from somewhere in here, and I'll show you that in a little bit, and then we compare those anadromous fish to freshwater fish. So you'll hear those terms a lot. Another reason why I like playing with stickleback, or studying stickleback, is because you can actually watch them develop live. And so what I have here are stickleback in chorions, and this is just watching a live fish through a flask. And what you can see here is the fish. Here's the fish egg. Here's the spine of the fish. This little thing is a beating heart. And here I'm just going through the microscope and showing you there's the tail. You can watch pigment appear. You can watch cells. Depending on the microscope you have, you can actually watch heart beating. You can watch blood flow. And you can watch this over time. So this is 3 days after we fertilize the eggs. That's 3 days post-fertilization. Here's 6 days post-fertilization. And at this point, you can see the eyes are a little more developed. There's more pigment. You can actually, in this video, I was able to show the blood flow. So if you look closer here, I'm going to zoom in. There we go. You can actually watch the blood flow over the egg yolk sac here, and then also within the fish. And again, there's the heartbeat. So you can watch these fish develop live over time. Oh, that one moved. They're alive. they're definitely moving. The other, and I spend way too much time doing this. But the other cool thing about that is if you tag these cells with some fluorescent protein, you can see where those fluorescent proteins are, those fluorescently tagged cells are going. And you can also watch the stages of development. So you can say, you can record when the eye appears, when pigment appears, when the vertebrae appear, etc. And I'm showing you 3 and 6 days post-fertilization, but we can watch this all the way from the single cell stage all the way through to about 3 weeks post-fertilization. We do that a lot in our lab. And this is just showing you that time series. Here's 0 days post-fertilization, 0 hours post-fertilization for age, all the way to 8 to 9 days post-fertilization. And as I said, we can take this out to a couple of weeks. We also get large clutches from single crosses. So if we take one female and we squeeze the eggs out of her, and we can do that and keep her alive, we get 200 eggs sometimes. And then we can cross them with, we can do an in vitro cross in the lab or in the field, and with sperm from a male, and so we can do these really nice genetic crosses. The juvenile are optically transparent, which I just talked about. The evolution and development is thoroughly described over well over 100 years. It shows a lot, it shares a lot of pathways with involved in host microbe interactions and immune responses with mammals, including humans. So this is a really nice series of studies that was done by David Kingsley et al. That you should look up if you're really interested in this. And what they showed is that a gene that was responsible for a phenotype in stickleback, these number of bony plates here, was also responsible for the regulation of that gene, was also responsible for the regulation of genes in humans that led to us developing the human foot shape that we have from a chimp foot shape, which is really cool. And again, our fish have natural genetic variation. I'm working with wild populations, so I go out and grab fish from the wild and bring them in the lab and play with them. It's really fun. We have lots of tools to examine host microbe interactions. So we have all the RNA, DNA sequencing tools, and in fact, some of these were developed in stickleback. So we've got some really nice tools to look at RNA and DNA. We can plate microbes, which we do a lot in our lab, so we can play with them later. We can count immune cells. As a postdoc, I counted them this way, which is long and painstaking. So I had a postdoc for a while, Lucas Kirschman, who's now in Kentucky, and he developed flow cytometry. And this was using a protocol from Natalie. Oh my gosh, Natalie. I am blinking on your last name. Natalie, who just started her own lab and is amazing, and you should talk to her, and I just completely blanked on her name. I'll have to look it up later. Anyway, Natalie developed a flow cytometry assay to count stickleback cells, and we use that in our lab. And then I had postdoc Emily Leskak, who was amazing, and she had previously studied behavior in stickleback, and so we adapted her behavior assays for juveniles. Okay. So, part 1, let's talk about who's there. In the corner of each slide, you're going to see a list of names, and these are the people who are working on these particular projects. So Ryan Lucas is a master's student in my lab currently. He's wrapping things up. Here's Emily Luskak, who I told you about before. Katie D'Amelio was a lab tech in my lab who is now working, well, she's in negotiations for a job with a government agency, and she was amazing. And then Patrice Timmons started out as an undergrad and is now a post-bac volunteer in the lab. So what they were working on is comparing microbes in the water to microbes in our fish. For those of you who aren't microbiologists, most of the microbes we deal with are not this big. I did that just to illustrate. And here I'm showing a fish that has a lot of different microbes, and those microbes most likely came from the water. And Ryan is comparing the microbes in the water to the microbes in the fish. And this is just to show you there's a whole lot of microbes in there. So he's doing this not for one population, but actually, what are we up to? 6 different populations over time. So this is a longitudinal study as well. He has that DNA and it's sequenced, and I was hoping we would have pretty graphs to show today, but well, you know, bioinformatics, you always run into issues. So we're hoping to have that information soon. But these are just the places that we've gone to. Again, these are all within driving distance. I think Big Lake is about an hour and a half from our campus, which is right here. So these are all places that we go to on a regular basis to grab fish and compare the microbes in the water to the microbes in the gut. And we also go to some other places. So this is a series of lakes up near Talkeetna, and they are really neat because they are, some of these are connected, and there's likely gene flow between the populations, and others are isolated. So we have a series of lakes that are in an area that should have some similar microbial community structures, but the populations of stickleback some have gene flow and others don't. So it's going to be a really fun series to look at. This is just showing you what this looks like. So we go out in the field, we grab some fish, we dissect them in the field often. Sometimes we take them back to the lab if it's below 0. And then this is showing the traps that we throw into the lakes. This is Ruth Eisenberg, a former student in the lab. And then we take the fish and the microbes and everything else back into the lab, and we do a bunch of bench work as well. So we do a combination of field work and bench work in our lab to look at these host microbe interactions. So Emily and I are really interested in figuring out, because we're doing wild work and lab work, if we could recapitulate the wild microbiota in the lab. So we did this fun little experiment where we looked at the microbiota of adult wild fish. Each one of these bars is a different fish, and each color is a different phyla that was identified in the gut of that fish by 16S sequencing. And you can see here that there is some variation from fish to fish that's totally normal and is what you see in every wild type of animal out there that's ever been sequenced. The other thing to note is that this particular group of fish has a lot of different microbes that are found in humans too, and I'll talk about that in a minute. And then we took our juveniles and we reared them in water that was spiked with water from the wild. And what we found is that at, I think this is 28 days post-fertilization, it might have been 2 months. I'll have to go back to the data and look. But these juveniles had very similar looking gut microbiota to the adults. This was done by Dr. Emily Luskak. And we found that there was no difference in the microbiota from the wild-caught adult fish and these juveniles that we had reared in the lab with microbiota, with microbiome that was collected from the wild and brought into the lab. So this is really cool. We can now recapitulate wild Alaskan stickleback microbiota in the lab. And then we use that information to figure out who was there. So in stickleback, we have a lot of the same microbiota as microbiota that's found in human gut microbiomes. So here we have a lot of, each one of these blue bars is a type of bacteria that's shared between stickleback and humans, and 2 of the many other model organisms out there, zebrafish and mouse. And so there are a lot of microbes that are found in stickleback guts that are also found in humans and found in some of these other models. And there are some that are unique to stickleback and some that are found in humans, but in that particular setting, we didn't find them in stickleback. So this is really cool. Now we have some way of recapitulating microbiota in the lab, and we may be able to colonize human microbiota in stickleback for future studies to figure out how human microbiota affects development of the host. So some of the other things that we do in the lab, we don't just look at the microbiota by DNA sequencing of the DNA. We also isolate live microbes, which is really fun. So here is a collection of different live microbes that we've isolated. These are all anaerobic microbes. They have lots of different cool phenotypes. I just love looking at these under the microscope. And fortunately, so does my undergraduate turned lab tech, Rachel Crimp. She isolated all of these anaerobic microbes and using this big anaerobic chamber that you can see here. She characterized the phenotype and then used sequencing to identify them. She's isolated, I think, 30 different microbes. I'm just showing you 6 of them here. Some of them are facultative anaerobes and others are obligate anaerobes, meaning that they cannot live in the presence of oxygen. And what's really neat about these obligate anaerobes is that they include Clostridium, which is a microbe that's known to colonize human guts and can also cause disease in humans. So this is exciting. We have a library of over 300 facultative and obligate aerobic and anaerobic microbes that we're constantly adding to. These are different phenotypes, and most of them are in different types of different groups, so different phyla. We identified those microbes by sequencing of the 16S gene, so targeting a bacterial-specific gene, but we had 20 microbes that didn't have any identification, that had a sequence that that wasn't found in the current databases. And so I had a student, an undergraduate student, Jessica Pinard, who knew how to do a lot of old school microbiology, and I had her run a bunch of assays, and these are pictures from her assays. So she ran, she took these 20 different isolates and she did some old school microbiology, including triple sugar iron agar, which looks at metabolism, starch assay, which looks at exoenzyme protein production, thioglycolate, which looks at the ability to survive in different oxygen different oxygen environments, Makani plate agar, which helps identify gram positives from gram negatives and tells you something about their metabolism, sim agar, which has to do with metabolism and also oxygen, and then TCC and MTM assays, which are used to look at metabolism, identify another pathway of metabolism, and look at mobility. So this is really cool. She's now taught these assays to 2 other undergrads in the lab, Sharon and Katherine, who started with me as freshmen and are now developing their own interesting projects. So Jessica took all that information and made some nice tables. I'm just showing you a handful of the isolates here, and she's used some dichotomous keys to identify the genus of different, some of these different microbes. What's neat is that some of these don't fit into any genuses. So these are microbes that haven't been characterized anywhere else. And we're in the process of writing these up now. What's also cool is that there were some fungi. I'm just showing one here that we think is a candida. This is interesting because I've never worked with fungi, and we didn't use agar that, we didn't use media that would typically target fungal species. So this is really neat. It's a new direction for the lab. So what do we do with all these microbes? We manipulate the microbes in the gut of these stickleback. We do notobiotic studies, noto meaning known, biotic meaning life. So we are able to characterize the, we do these studies where we manipulate the microbes that are associated with the gut and associated with the fish as a whole, not just the gut. So we can make fish germ-free. Here's our little stickleback. And we can take fish from the egg stage and fertilize them, and then when they're at about the 4 to 8 cell stage, we take those fish and sterilize the surface of them using iodine, which is PVPI, and bleach, and then we can add back microbes. So usually we do this about 10 days post-fertilization. That's when the gut opens, but we can do it whenever we want in here. And then we collect the fish sometime around 14 days post-fertilization. We can add back conventional complex communities of microbes that typically involve hundreds of different types of microbes. And we do this just by taking some water, either from the wild or from one of our tanks that contains adults, and spike our germ-free fish with them. We can also add back individual microbes. We have that library of 300 different microbes. We can take any of those microbes, grow them up in vitro, and add them to the water to see how that particular microbe manipulates the development of the host. Or we can make mock communities where we take specific members and add them to the fish. And we know exactly which microbes are going in and what ratio they're going in. I just wanted to show you what that looks like. So we make the fish germ-free in the hood, and then we transfer them to these flasks and raise them in an incubator. Now, I told you that Rachel Cramp is doing some fun work with these anaerobic microbes. One of the things that she did last summer, and she's going to repeat this summer, is she took some of her one of her anaerobic isolates and an aerobic isolate and inoculated the fish water with both the anaerobic and the aerobic fish, or anaerobic and aerobic bacteria, and then took the fish at 14 days post-fertilization, removed the guts, and plated the anterior and the posterior part of the gut. And what she was trying to figure out is where the anaerobic microbes go versus where the aerobic microbes go. This is really to understand colonization dynamics. Then she counted all of the different microbes that were there. These microbes have different phenotypes, so it's hard to see here, but underneath these black dots, there are some really small microbes. Those were the anaerobic microbes, and these big blobs are the aerobic microbes. So two different phenotypes we can easily identify on a plate. And what she saw is that the anterior part of the gut had more of the facultative bacteria, and then the posterior part of the gut had more facultative bacteria, and the anterior of the gut, so the part that's closest to the anal vent, had more anaerobic bacteria. I just lost my train of thought. Sorry about that. The front part of the gut, can I hit rewind? The front part of the gut, the anterior part of the gut, which is closest to the stomach, had more aerobic bacteria, and the posterior part of the gut, which is closest to the anal vent, had more anaerobic bacteria. And that's what we had hypothesized, because we think that the posterior part of the gut is a more anaerobic environment. We don't have a way to identify the oxygen levels in the gut right now, but we had hypothesized that the posterior part of the gut had less oxygen, and that we would find more anaerobic bacteria there, and that's what she found. So this is a really low number. We didn't have a ton of fish in this particular experiment, so we're going to be repeating this, and we're going to be repeating it with fluorescent microbes. We have a really great protocol from Travis Wiles at the University of Oregon, and this protocol is to make any microbe fluorescent. And as I said, of our 300 species, or our 300 different microbe isolates, a bunch of them are unknown, a bunch of them have never been manipulated in the laboratory before. So we took Travis's, Dr. Wiles' protocol, and made a bunch of our microbes fluorescent, and that's work being done by Sharon and Katherine under the guidance of Katie. And Rachel Cramp has done this with her anaerobic microbes as well. And then what we can do is basically make fluorescent poop. We can add these fluorescent microbes to the gut. This is the gut in a live fish, and you can see that it's colonized with with fluorescent microbes. When you anesthetize the fish, some of that comes out, so we make fluorescent poop in the lab. It's a lot of fun. And if you have a high-powered microscope, then you can also see where these microbes are going. You can watch them interact with each other. You can watch movement, all the fun things that you can do with fluorescent microbes. Okay, so those are ongoing experiments. So now I'm going to tell you about some of the really fun things that we're doing manipulating microbiota. So part 2 is what happens to the host when the microbiota is disrupted. I'm going to tell you about our germ-free fish and results that Dr. Emily Luskak is writing up. I'm not going to talk about our environmental contaminants, but I have a few different projects going in the lab, one looking at perchlorate, one looking at fluoridone, one looking at crude oil, and those are all being run, the crude oil and the fluoridone are being run by undergraduates, and the perchlorate was being led by Emily Luskak, and now somebody else. If we get funding, we'll take that project over. And I'll talk about our antibiotic disruption. So, as I said, the fish are transparent. So here we have a juvenile fish. We can do a lot of different measurements with these transparent fish. Our typical go-to measurements are eye diameter, swim bladder, and swim length, and then standard length from the tip of the nose to the tip of the tail. We can also look at somatic development. That's just the measurements I showed you here. We can look at behavior, and we can also characterize the gut microbiota, diversity, and membership. A lot of this early work, all of the early work was led by Dr. Emily Luskak, and she's writing up all this information. One of the early students that was helping us with measurements was Keegan Whitlock. So what happens if you take a fish and raise it in the absence of microbes? We did that. We took fish and raised them in the presence of complex microbial communities, which is conventional, or we raised them in the absence of any microbes. That's germ-free. And we did all the measurements that we could do. We looked, well, the measurements we have good measurements for. So we looked at swim bladder area, for example. In our conventional fish, we saw that anadromous fish had a bigger swim bladder area compared to our germ-free fish. And that was true for standard length. This is, sometimes we see a difference in standard length between our germ-free and conventional, and other times we don't. In this particular assay for these anadromous fish, and this one population, we saw a difference in the standard length. Emily also does a lot of behavioral assays. So she takes a flask that looks like this and finds every fish in that flask and then follows their movement over time. And so we take a video of about 5 minutes and then watch where each individual fish goes. We look at total number of squares. So how many squares did this fish move into? How many of those squares were unique? So did they just go back and forth between here, or did they go through many different squares? And then how much time did they spend on the parameter? Because in other labs, they've shown that that has to do with how bold these fish are. If they spend a lot of time on the parameter, they're not very exploratory, and they're referred to as more shy. But if they come into the center where there's more likely to be predators or things like that, then they're considered to be more bold. And she had help with from Haley Miller and Kiana Verplank, 2 undergrads in the lab. She took all the behavioral information and did principal component analysis. So this is the PCOA plot showing that conventional fish, seen over here, these 3 dots, had different behavior than our germ-free fish, these 2 fish over here. And this was an anadromous fish. And the difference in these germ-free versus conventional fish was driven by differences in parameter squares, so how much time they spend on the edges, and germ-free fish tended to spend more time on the parameter. Now, we didn't just look at anadromous fish, and we didn't just look at that one particular aspect. We also looked at nearest neighbor distance, which is how close one fish is to other fish. And this was work done again by Kiana. And she found that the anadromous fish had different behavioral dynamics than the bootleg fish. So here we have 2 different assays run on 2 different days. And in this assay, the rabbit suit, and in this assay, the fish that were raised germ-free shoal less, so spent less time with close to other fish than the conventional fish, the fish raised with the conventional microbiota. And here we have in bootleg some really big wampum error bars here. And it turns out that the Boot Lake fish, these freshwater genetically variant fish, have that don't appear to have a difference in their shoaling behavior between the conventional and germ-free environments when raised in the conventional germ-free environments. So that's really cool. Now we have a gene by environment interaction and behavior in this specific behavioral dynamic, which is how much time or how close are these fish to other fish. We've done these experiments with a couple of different populations now, freshwater and anadromous, and from the experiments that I showed you, we saw that anadromous fish had decreased exploratory behavior, smaller swim bladder, and smaller size overall, and the freshwater fish were more exploratory. And for both of these populations in the experiments shown, the germ-free fish in both freshwater and anadromous time spent more time on the parameter, had decreased shoaling, and there were no effects on survival. So this is cool. Now we have a gene by environment interaction on different behavior and developmental assays. And as I said, she's looked at several different freshwater and anadromous populations. So she's in, Emily, Dr. Luskak is in the process of writing this information up now. We also did some antibiotic manipulation experiments. So Emily had funding from NSF to examine how chronic exposure to low levels of antibiotics affected the development of these fish from different populations, and this mimics levels that we would see in wild fish habitats. And then Ryan Lucas, who's a graduate student in my lab, for his first semester, or for his first year, he had funding to look at the short-term clinically relevant levels of antibiotics that are similar to levels that kids are exposed to early in development. And this was funded by INBRI. And the idea there is humans are exposed to a large amount of antibiotics for a very short amount of time during a critical stage of development when they're kids. And so we were curious as to how different populations would respond to that short-term exposure. So to do these assays, what we did is we took 3 different populations of fish, 2 freshwater fish, and 1 anadromous population of fish. And then we exposed them to low concentration or high concentrations of an antibiotic, in this case tetracycline, or ethanol, which is the solvent used to dissolve that antibiotic before adding it to the water. We did these in triplicate, and we added antibiotics shortly after the gut opens in conventionally raised fish. And then we collected the fish early in development, 2 weeks post-fertilization or 14 days post-fertilization, and looked at those early stages of development. Emily had plans to do chronic exposures for a year, but she has moved on to bigger and better and a very awesome job. So if anybody's interested in looking at chronic antibiotic exposure, come talk to me. Okay, so what did we find? We found that the anadromous population had very little response to this change in microbiota. So here's our control fish. Here's fish raised with a little bit of ethanol that matches the amount of ethanol they would have gotten if they had been, if antibiotics had been added to the water. And here's our low and high concentrations of antibiotics. No difference in standard length. But we didn't just look at one population, right? We looked at a couple of different populations. So this freshwater population had some decreased growth in, looks a little bit shorter in our ethanol exposed fish, which is really interesting because that was a very tiny amount of ethanol, and possibly some difference in low and high concentrations of TET, but it's really, there wasn't a ton of difference overall, especially when you compare them to the other freshwater populations. So here's our control and our ethanol again, and here is our low and high concentration of TET. And wow, was there a difference in the growth of these fish? So this is really cool. This is similar to what farmers see when they expose animals to antibiotics. It's one of the reasons that farmers give antibiotics to animals is so that they will grow bigger faster. And that's what we saw in one population of our fish, but not the two others. So here we have a gene by environment interaction in development when these fish are exposed to different concentrations of antibiotics. Super cool. We also measured several other different measurements, and I'm not going to talk about all of them, but what was interesting is that the swim bladder didn't have the same differences that we saw in the standard length and the eye diameter. So different regulatory systems for swim bladder development compared to the eye diameter and overall length. So the conclusion from that experiment was a gene by environment interaction appeared to drive responses to antibiotic exposure, in that one freshwater population had developmental instability when the microbiota was disrupted by antibiotics, compared to the other freshwater or the oceanic anadromous population, and there was no influence on behavior or survival. So that was really interesting. We expected to see a difference in behavior. We saw no difference in behavior. So as I said, Emily is in the process of writing up this manuscript, and we hope to have that out soon. In the interest of time, because I want to make sure I get to this last story that's really new, I'm going to skip over the mock community analysis that our former student, our student Kenny did, our former undergrad, now master's student. But this is also work that we're writing up now, in which we inoculated fish with different mock communities, disrupted that mock community with antibiotics, and saw a difference in development. So it's a cool project. We're writing it up. But I wanted to get to this last project that was done by, the last project that I'll talk about, was being done by, that was done by a former postdoc, Lucas Kirchman, who's now in Kentucky, and a medical student, Anastasia Katchanova. And this is a really fun project that I've been wanting to do for, since I became a PI here. And Lucas and Anastasia just happened to be really excited about it as well. So I finally found somebody in the lab who would do this. So this is a really cool project. What we did is we took two different populations, an anadromous and a freshwater population, and we made them germ-free. Half of those fish, we took all the fish and made them germ-free, and to half of them we added back a conventional microbiota, so that complex microbial community. We did that at 6 days post-fertilization. And then, this is really fun, Lucas and Anastasia exposed half of the fish from each of those groups to Vibrio. And Vibrio is, and we took a Vibrio species that has been shown to be pathogenic in fish. Now, this is the first time we had associated stickleback with this particular strain, and we already had all these compounding factors, and so we only did one concentration of Vibrio. In hindsight, I think in the future when we do this, we'll use a higher concentration of Vibrio. But we took some Vibrio and added it to our conventional and germ-free fish, and then looked at development at 14 days post-fertilization. These fish were infected at 11 days post-fertilization. So again, this is a potential pathogen for fish, so we added to conventional and germ-free fish from two different populations to look at a gene by environment interaction and the ability of the microbiota to protect the fish from Vibrio exposure. Really complex question. I've been wanting to do it forever. Huge kudos to Dr. Kirchman and future Dr. Katchanova for finally doing this project. And again, Lucas is writing this up. So many manuscripts that we're working on. So what they did is they measured survival, immune gene expression, development, and behavior. I'm going to start with the development. So Lucas and Anastasia went through every single fish and looked at several different lengths, several different somatic development measurements. This one is snout to vent, anal vent length, and that is just another way to look at overall growth. And what they found is that neither genetic background, microbiota treatment, or pathogen exposure had an effect on growth. This is cool because we expected Vibrio to affect overall growth, and it did not. Here's our anadromous population. Here's our freshwater population. The uninfected is in gray. The infected is in red. These are conventionally raised fish. These are germ-free. Conventional, germ-free. No difference. Super cool. You wouldn't think that negative results would be cool. I thought they were super cool. So we also looked at several other individual measurements. So here's eye diameter. And in our anadromous fish, there didn't seem to be an effect either by conventional versus germ-free or Vibrio uninfected versus infected. In fact, stickleback from the rabbit slough had significantly smaller eye diameter relative to their SBL and compared to Westchester Lagoon. So this was interesting because the Westchester Lagoon, the freshwater fish, when they're adults, tend to be much smaller than the anadromous fish. And here at 14 days post-fertilization, we had much larger freshwater fish in the eye diameter than we did our anadromous fish. And what was interesting is that, again, overall, this measurement of overall length, this snout to vent length, and he did do overall length as well, there was no difference. So in one specific measurement, eye diameter, there was a difference between Westchester Lagoon, our freshwater population, and our anadromous population. And interestingly, this is all interesting. I love this experiment. The uninfected fish had a larger eye diameter compared to the infected fish. Again, no difference in length, but a difference in eye diameter, indicating that the exposure to Vibrio affected one particular measurement of development different than another developmental marker, and differently in germ-free versus conventional fish, and differently between anadromous and freshwater. So lots of interactions going on. They also looked at swim bladder length and saw that stickleback from both populations had similar swim bladder lengths, so different than eye diameter, and that stickleback from rabbit slough exhibited shorter swim bladder lengths when it raised germ-free and exposed to Vibrio. So you had to have both things, germ-free and exposed to Vibrio. Big difference there too. That was fun. Overall swim bladder area, we saw the same thing. So same patterns. But interestingly, Westchester Lagoon freshwater fish that were raised germ-free had smaller overall swim bladder area than the conventionally raised. So that was interesting because overall length, not much of a difference. Area, a bigger difference. So they, that's really neat, and we're going to follow up on that. So just to summarize the overall growth, there was no difference in overall growth in conventional versus germ-free, anadromous versus freshwater, but there were different gene by environment responses and development between anadromous and freshwater fish, both between the microbiota that they were exposed to, whether it was conventional or germ-free, but also whether they were exposed to Vibrio or not exposed to Vibrio. So cool stuff. Cool. And then we also did some immune work, and this is work done by Lucas again and Anastasia, but then also Kelly Ireland, who was a former undergrad who's now a grad student. She and Lucas developed this, they adapted a QRT-PCR assay for several different immune genes from a McCall et al. Paper, and they we used that QRT-PCR assay to measure transcripts in our fish. These are really tiny fish, so this is overall, over the entire body, how much of a difference is there in the transcript level. And I'm showing you TNF-alpha first, but we did a couple of others as well. So if you expose fish to any fish from either population to Vibrio, we saw a decrease in TNF-alpha relative normalized quantity. This is cool. TNF-alpha is a pro-inflammatory cytokine that's made by immune cells. And the Vibrio exposure decreased TNF-alpha expression in all of the fish, whether they were raised germ-free or conventional, just overall, like if you group all the fish into either uninfected or infected, and using that just as a variable, we saw a difference. And then we also saw that rabbit slough had higher expression of TNF-alpha than Westchester Lagoon. I had done immune assays as a postdoc and saw a difference between anadromous and freshwater populations. So this is cool. In TNF-alpha, again, we saw a difference in expression of a specific gene. And we also found that Westchester Lagoon fish raised germ-free had increased expression of TNF-alpha. That's opposite of the immune responses that I saw as a postdoc using a completely different freshwater population and using a different measurement of immune cells. So this was, or immune response, so this was a really cool finding. We saw no genetic background microbiota or Vibrio effects on another gene, pro-inflammatory cytokine, IL beta. So this is exciting. I'm really excited. Lucas and Kelly and Anastasia are working to write this up now. So as I said, we grouped fish by germ-free, conventional, Vibrio exposed, not exposed, and 2 different populations. And so Lucas ran the analysis looking at just conventional and germ-free, infected and uninfected, and broken down by population, and saw that Vibrio exposure decreased TNF-alpha expression in all fish, which is what we saw before. But that rabbit slew fish had higher expression of TNF-alpha overall, which is what we saw before. And that Westchester Lagoon fish raised germ-free had increased expression of TNF-alpha. This is different than other germ-free conditions in other immune systems or in other model organisms. So this is really cool. Germ-free conditions typically are associated with impaired innate immune responses. So we expect the germ-free fish to have decreased inflammation. And in anadromous fish, we don't see that. In our germ-free freshwater fish, we actually see increased pro-inflammatory cytokines. So this is fun. Oh my gosh, that is so much information. And I didn't get to some of the other projects that we're doing. Looking at the interaction between evolution and microbiota is another project that we have going on with Mike Bell and Krishna Virama. With Dan Bolnick, we've established a stickleback stock center that we hope to have running by this spring so that people can tell us that they want fish and we can send them out to them. And that's a 5 PI, Dan Bolnick is a 5 PI grant from NSF. Dan Bolnick's the lead on that. We've been working with Brandon Briggs a lot on the water versus gut microbiota stuff because Brandon is really great at identifying microbial communities in low biomass areas like volcanoes and water and extreme environments. So we've been working with him a lot. Frank Von Hippel and Pat Tomko, I didn't get to talk about our fluoridone and perchlorate exposure experiments, but we're doing some fun contaminant experiments with them as well. I have funding. Thank you so much, NIH. Thank you so much, NSF. Thank you to INBRI, who is who initially funded that pathogen exposure experiment that we did. Thanks to NSF for funding Dr. Luskak when she was in my lab. We've gotten funding from AFS and UAA, and American Heart Association or American Heart Institute is down here and got cut off. These are the people in my lab. This is why I'm able to get so much done. I've been really lucky. I've had some amazing postdocs. I've had 3 postdocs. Shout out to Dr. Nashoba, who did a lot of DNA work that I wasn't able to talk about here today. I have 4 graduate students right now who are pretty awesome. Anastasia was a med student. I've had a bunch of lab techs. They tend to come from post-bac backgrounds where they're interested in figuring out what they want to do next, and they spend a year with me. So thank you to my awesome technicians. I have had some post-bac volunteers. I'm showing Tyler here, but Patrice is now a post-bac volunteer in my lab. I have a ton of undergrads, and I've also had high school students. So, you know, for those of you who are at different stages in your research career, and you're not sure if research is something that you can do, you can start in high school and work in a lab through various different funding opportunities. So keep that in mind. If you're somebody who's an undergrad or a grad student now or a PI, and you know of a high school student who's really interested in working in the lab, bring them in. and show them what it's like to work in a lab. I think it's a really valuable experience. So that's it. That's what I have.
Awesome work.
Thank you.
So thank you very much again, and thanks for bearing with us through the technical difficulties. I do have a question. So the thing that struck me was the growth differences. So the antibiotics make one of the populations grow bigger, and then they don't make the other ones, right?
Yeah,
So
right.
are you trying to chase down the genetic basis of that? Because that seems like the holy grail of like what people are trying to get at, and it seems like a system you can go for.
Yeah, I am really excited about that work. It's just, this is what the one that you're talking about. I'm really excited about that work. And so Emily's left the lab, but I have other students who are interested, or I'm hoping to have other students in the future who are interested. I'm not an evolutionary biologist. I adapted this evolutionary model, but I am not an evolutionary biologist. So I'm having fun collaborating with people like Krishna and Mike Bell, who are geneticists, to track down those questions. And so or track down those answers. So that's something that I'm going to collaborate with a true geneticist in the future. And you know, Bill Cresco, his lab, that's one of the things they do. They look for gene, they do, what do you call them? GWAS studies to identify these genetic factors driving these really cool phenotypes. So he's another person I want to collaborate in the future to identify the genes involved.
Cool. Let me just cruise around and see if there's anything any questions. Mark Martin wants to join your lab, so I will point that out. He says he wish he could start over again.
Well, while Dave does that, I have a question for you, Kat. So I was excited about the data that you were sort of suggesting that the first part of the gut, what is that? The proximal part of the gut?
Anterior.
The anterior, there we go. The anterior was more aerobic than the posterior gut in your fish. And I'm on a students committee who's looking at fish microbiome work. It's not really my area, but we were talking about, you know, how anaerobic are fish guts, and does that change at different times over development? And you know, I had some wild ideas that all sounded very hard for how to actually measure that and determine that in vivo. But I was wondering if you had ideas about, you know, first of all, you know, reasons why the anterior gut might be more aerobic than the posterior, and also ways to get at that type of question.
So we came up with a hypothesis that the anterior gut would be more aerobic than the posterior gut, and the posterior gut would be more anaerobic, so less oxygen, because that's what we see in a lot of other organisms.
Yeah.
Other organisms, you see that there are more anaerobic microbes in the posterior part. And Rachel and I have been talking about different ways to measure that. There was, I looked into microprobes because you can buy these really, we have a physiologist in the department who looks at heart cells in turtles, and he has these really tiny probes. We can measure things like that in small environments, but we haven't gotten that to work in stickleback. We haven't pursued it. One of the other things I'm thinking about is there are some proteins that'll turn on in anaerobic environments. So fluorescently tag one of those, throw it in, and see if it turns on in the posterior part of the gut versus the anterior. And you can do that in fish because you can see through them at early stages. That's true for zebrafish and true for stickleback. So I think this is a great system to figure that out. And I also want to see about like an oxygen gradient, right? Because, you know, we talk about anterior and posterior guts as though there are these two very distinct things, and there's a sphincter in between them. So there's a closing between the anterior and the posterior gut. But what if there's an oxygen gradient like leading up to that closure? And I just think it would be really fun to explore.
Yeah. Do you know if those protein probes, can you just throw those in, or are they things that are expressed in vivo by some sort of microbe that you're feeding in? Or
I would do it in a microbe.
Yeah.
I would do it in a microbe. So I would take a microbe that I know can survive in the posterior gut, which, hey, we just happen to have, and throw a fluorescent protein behind a gene that's driven by oxygen deprivation. And we have facultative anaerobic microbes that should be able to survive in both the posterior and the anterior and the posterior part of the gut.
Yeah, cool.